Hello and welcome to the Immune Epitope database. My name is Nina Blazeska and I'm the Project Manager of the IEDB. Today we'll be showing you a brief demonstration of how you can use the IEDB for your SARS-CoV-2 research, and in particular the ImmunomeBrowser functionality. Shortly you'll be hearing from Dr. Alba Grifoni on the T-cell data available, which will be followed by an overview of the B-cell data presented by myself. As always, we welcome feedback and suggestions from our users, so feel free to contact us via email at help@iedb.org. We'd also like to take this opportunity to thank you for your interest in the IEDB, and we hope you find this video useful.
Welcome to this tutorial on finding T-cells responses in SARS-CoV-2. My name is Alba Grifoni, and I'm going to guide you through how to retrieve this information in the IEDB database. So the IEDB database allows you to retrieve different information about epitopes. And in order to focus our research on SARS-CoV-2, we have to type this information in the organism section. So we're going to have SARS-CoV-2. You just need to type the beginning of the name of the virus, and then it's self-complete and allows you to retrieve the information you're interested in. If of interest, if you want to focus your query not only to the entire organism, but to specific antigen, you can. For today's purpose, I'm not going to do that. And considering the fact that the different epitopes have been retrieved by different animal species, the IEDB allows you to select by the host of interest. And today I'm going to show you the data coming from human T-cell responses. And as the main purpose of this research is to actually look into T-cell response, we're also going to select our assay as T-cells. And you have the possibility to look only at the epitopes, so the positive response that you are retrieving, or you can also look at both. And the reason why this option is there is because sometimes you want to understand if a specific portion of the virus has been tested and been negative or has never been tested at all. So for the purpose of the current query, I'm going to just keep the positive response, but just so you know, you can look at both. So when we are interested in looking at the T-cell response for CD4 and CD8 T-cell response, it's important to notice that clearly the MHC restriction is different. So if we are interested in retrieving information from all the T-cells, we don't specify the restriction. However, if we want to focus our query on CD4 or CD8 response, we're going to select class 1 for the CD8 and class 2 for the CD4. So let's gonna look at the CD8 response first and just press the button search to actually retrieve the query. And the results that we see here show us that there are 1,149 epitopes currently available. If we don't remember what query we did, IEDB is smart enough to remind us what were the original filter that were applied. We do see that these epitopes are actually compiled based on different ways. For instance, if we want to look at the epitopes in the context of the assay, we can. So each epitope is described pertaining to the assay, pertaining to the MHC restriction, and also pertaining to the specific reference where this epitope has been isolated. We also have information about the receptor, and we do have a full list of the references where these epitopes have been derived in IEDB. And if we're interested in looking more in depth about the specific reference, the IEDB also provides the PMID or the reference ID in case the paper is not published yet, but is available in bioRxiv. And this is an important point, particularly for COVID-19, as we know that the research is progressing so fast, and IEDB is actually keeping up with that and try to include all the possible information available. Another way it is interesting to look at the data is to understand how these epitopes map in the different antigen of the virus. So IEDB allows you to do that by clicking on the antigen section. And indeed, when we look at the different antigens, we do see how many epitopes pertain to different antigen of interest. We want to look at where are these epitopes positioned in the protein. We can do so by using ImmunomeBrowser. So the ImmunomeBrowser allows you to look at and appreciate the entire activity against the protein, what has been tested and not. And the results are actually plotted in different ways. In the first case, we look at the response frequency. So we're looking at how many positive donors are found in the specific epitope region. And what you see over here is the confidence interval with the lower and upper bound. By clicking on a different position, we can actually appreciate the specific numbers as well as the position in the sequence. A different way to look at this data is to look at the epitope assay counts. So here, we're really appreciating how many times these epitopes were found positive or negative. Now, in this specific case, if you recall in the query, we specifically select for positive response, which is why, of course, we don't see any negative. But what we can appreciate by looking at the different position, we have for each position the number of positive and negative donors. So in the context of SARS, for instance, if we are interested in a specific position that perhaps is mutated in one of the emerging SARS-CoV-2 variants, we can do so by looking at this information. And one interesting point that we observe here is that in the CD8 response, we do see a more homogeneous recognition throughout the spike protein, while in the context of the CD4, there are more defined immunodominant regions. So if we want to look at this, what we can do is to go back to our original query. And instead of going back to the original homepage, we can just modify our query by selecting the CD4 response in this case, so the class 2 restriction. You can change multiple parameters. What is important, and once you do so, you press the search button. So that allows the database to actually update the query that you are asking for. And in fact, you see that the number of epitopes now has changed, and there are only 420 epitopes pertaining to the CD4 response. If we want to look again to the ImmunoBrowser on the spike protein, now, as you can see, the picture has changed, and you do see different immunodominant region, and actually the region locating on the RBD domains is actually not as recognized as observed in the context of the CD8 response. Whether you're looking at the CD4 or at the CD8 response, you can also extract this data in a tabular format. Therefore, we can look at the different sequence of the epitopes. We can refer to the single query of this epitope. And when we do so, we actually have the specific description, the type of assay that has been used to actually identify these epitopes, as well as external link that refers to additional tools of the IEDB that can be used based on the initial query for this epitope. Going back to our original query, what we can see is that together with the epitope sequence, we also have an information about the map position. We have an idea about the identity. And when we talk about the sequence identity, in the context of the ImmunoBrowser embedded in IEDB, they use as a reference the ancestral Wuhan strain. If there is the interest to look at your own list of epitopes or your own list of sequence, then the IEDB provide a tool that is similar to the ImmunoBrowser we're showing, but it's a standalone tool that allows you to customize the list of epitopes that you want to include and also the sequence you want to map to visualize them. And with that one, I would like to thank you for your attention and thank you for following this tutorial. Bye.
Hello and welcome to the Immune Epitope database. My name is Nina Blazeska and I'm the project manager of the IEDB. In this demonstration video, I'll be showing you a step-by-step process of how to access the ImmunomeBrowser for B-cell data for SARS-CoV-2 epitopes, focusing on the spike protein. To start, we'll locate the epitope source panel on the IEDB homepage and select our organism of choice. So here I'll type in SARS, and as you can see, a series of related organisms are populated in the dropdown based on community ontologies. Now I'll select SARS-CoV-2. You're also able to select an antigen, but for purposes of this tutorial, we will keep this blank. Next, we can select our assays of interest, and for this tutorial, we'll be looking at B-cell assays. You're also able to select your assay outcome of choice. So positive data means when in the assay, the B-cell recognizes that epitope and generates a response against it, whilst negative means the B-cell does not generate a response against that epitope. So to understand the ImmunomeBrowser better, we'll select both positive and negative. And lastly, as we are focusing on SARS-CoV-2, we'll only be looking at human data, so I'll select that in the host panel. I'll keep all my other parameters the same and hit the green search. And as that's loading, you can see that the B cell view has been selected in our filter options based on our original query, and all my other query parameters are populated at the top. And now you can see all of the carefully curated B cell data available for SARS-CoV-2. As you can see, we have many different tabs with various results. So the epitope, antigen, assay, receptor, and references tab. And you can also export your results here. In the next step, we'll be focusing on the ImmunomeBrowser, which can be found by clicking on the Antigens tab. And this is just a pop-up reminding us of where the ImmunomeBrowser can be found. So as a result, I'll click on this histogram-like button. And now this is taking me to the ImmunomeBrowser. So at the top here, we have the name of the protein we are looking into. So SARS-CoV-2 spike glycoprotein. Next to it, there is also a link to the UniProt database, which has the protein sequence data, which we used as a reference to align with our protein. As you can see, there are also two graphs here of interest. So let's talk about this first graph, response frequency, or RF. So this is a graph informing us of how immunogenic a specific residue is in the analyzed protein. So RF equals R on N, where R is the number of subjects responded and N is the number of subjects tested. The x-axis here shows the values that are the residue numbers of the protein, while the y-axis indicates the response frequencies. What we see here is actually a confidence interval of the response frequency. This is very important because it allows us to correct for the sample size. Otherwise, a peptide found to be positive in only one donor, so R equals 1, N equals 1, would be considered more immunogenic than a peptide showing response in 99 out of 100 donors, as the former has an RF score of 1 and the latter of 0.99. So this graph is showing a range of possible values that will contain the real response frequency value 95% of the time if the same experiment is repeated an infinite number of times under the exact same conditions. Of course, we'll have different values for B cell and T cell data, so it is expected that the response frequency will differ even if we compare this with the same protein. So you can also export the results that are used to plot this graph here, and you can download or save the data in various file formats. Moving down to the epitope assay counts graph. So this shows how many times a specific amino acid is present in a positive or negative assay. So this graph gives us an idea of the immunogenicity from that region. And similarly, you can also download or save this data. Important to note that blue line indicates the positive data, whilst the red line indicates the negative. And then moving down to our table, this table shows every epitope that has an assay related to it. So that first column, epitope ID, contains the IEDB accession number of the epitopes, while the second column contains the exact epitope sequence. And as you can see, we have both linear sequences and discontinuous sequences. In the third column, we have the mapped position, which indicates where that epitope is located in the reference protein. And this is followed by the percent sequence identity between the epitope and the corresponding region in the reference protein. The next columns relate to subjects tested, responded, and the assay outcome. And lastly, the last column is the response frequency when we have a confidence interval of 95%. Now I'd like to dig a little deeper. We'd like to see what regions in the SARS-CoV-2 spike glycoprotein are the most immunogenic. So I'll just hover over the graphs here, but residues 13, just around here, to residue 303, which is around here. This comprises the N-terminal domain. Next, I'd like to zoom in on a particular area. So residues 319, which is just around here, to 541, which is just below this area, zooming in here. This makes up the receptor binding domain, or the RBD. Within the RBD, there's a particular area called the receptor binding motif, and this is from residue 437 which is just about here, to residue 508, which is just around here. And now I'd like to zoom back out. From these two plots, we see that the RBD domain is just one such area of interest. And apart from this, there are a few other areas that are fairly immunogenic. So again, I'll hover, but some examples that we have, residues of 530. So we're looking at around about here to residue 600. That area is quite immunogenic. We see from 612, which is just around here, to 659 here. That area is quite immunogenic. And in fact, the residue D614, which is frequently mutated in the variants, is located in this immunogenic region. And then lastly, region 760, which is about here, to 840, about there. That is another immunogenic region. So next, we'd like to see where mutations are observed in some of the circulating SARS-CoV-2 variants within these immunogenic regions. So to make this a bit easier, I'll be zooming in on a region about here to here. So for example, we have at position 484, we have a mutation E484K, which is observed in the following four variants: P.1, B.1.351, B.1.1.7, and B.1.617. And this is obviously situated in a highly immunogenic region and can thus alter antibody responses. Similarly, we have another frequently occurring mutation at position 501, which is just around here. And this mutation is N501Y, which is present in variants P.1, B.1.351, and B.1.1.7. Again, another highly immunogenic region. I have a few other examples to show you. So at position 452, we have L452R mutation, which is present in B.1.617, which was first detected in India, as well as two variants here in California, B.1.427 and B.1.429. And this is all, of course, at the time of recording this video. And then lastly, at position 417, we have mutation K417N, which is present in the B.1.351 variant, first detected in South Africa. So hopefully these examples demonstrate how one can use this information to assess whether the mutations in the variants are likely to affect the antibody responses to SARS-CoV-2 by determining whether they are located in highly immunogenic regions or not. As this demonstration, we hope, has shown, the IEDB can provide some really useful information about currently available SARS-CoV-2 data, which can be analysed to obtain some important insights. As more data continues to be deposited into the database, the results in the ImmunomeBrowser will, of course, grow richer. We also aim to add some new features to the ImmunomeBrowser in the future to continue enhancing that user experience and of course, the usefulness of these graphs for your research. But for now, we hope you enjoyed this demonstration, and thank you very much.